Yes, perfect.
So, thank you for having me here today. So, epigenetics is one of the more challenging topics to understand for even us as geneticists, and but I'm trying to going to try to simplify this topic so that we have an overall idea of what this epigenetics is and how it has a role in infertility, and what are the interaction of environment of genetics with infertility and its outcomes. So we were just discussing before this talk really started that some of our senior colleagues were doing yoga before the talk, and there was some exercise happening. So yoga so yoga positively regulates epigenomics. So now, to understand what epigenomics is, first we need to understand what genomics is and what is a genome. So when we talk about the genome, it is a total amalgamation of all the chromosomes that are present in our body. Each cell contains 46 chromosomes, and there are 22 pairs of autosomes and one pair of sex chromosomes, which could be an XX or an XY. So a genome consists of all these chromosomes. Each chromosome then has on it a lot of genes that are present. The gene has a coding region, and also there are intergenic regions that are present. So amalgamation of all the genes which are present on the chromosome and all the chromosome together is what is known as the genome. Now, we are still talking about genome and genetics. When there are changes in these genome, these are known as genetic changes. And the genetic changes are what we read in obstetrics, in gynecology, when there is a deletion in part of the genome or there is a mutation. When we talk about the chromosome, one of the chromosome we get from our dad or from the father, and one chromosome we get from the mom or the mother. So when we do not receive enough number of chromosomes and there is a missing chromosome, this is known as a deletion of chromosome. For example, in Turner syndrome, there is a missing X chromosome. So in Turner syndrome, there is an abnormality because there is a chromosomal disorder. That means one part of the chromosome is missing. Whereas in Down syndrome, instead of getting one chromosome each from the mother and the father, we get an extra chromosome from the mother. We get both the chromosomes of the mother and one from the father. So that's why there's an additional chromosome, and this is known as a trisomy. Whereas in Turner syndrome, it is known as a monosomy. So the first block of changes occurs at the level of chromosomes, and those are known as chromosomal genetic changes. Now, on the chromosomes, on each chromosome, there is a pearl which is present, and these pearls are a pearl of genes that are present. Now, genes are made up of these four nucleotides, the A, T, G, and C. Mutation or changes in the genes occur when any pearl of this necklace is replaced. For example, if the pearl A is replaced by the pearl G, or the pearl T is replaced by a pearl C. These are the changes on the genes. Now, these genes are present on the chromosomes. These changes which are present on the genes are different from the changes on the chromosomes, and these are known as mutations when one pearl is replaced by another pearl. So this is the second level of changes. The first is at the level of chromosomes, and the second is at level of genes, where there is a change in the sequence of the gene. This is the sequence of the gene, the pearl I was referring to, and there is a change in the pearl T to the pearl C, and this is the beta globin gene, and this change is causing beta thalassemia. So this is another disorder which is different from Turner and Down syndrome, which are chromosomal disorder. This is known as a monogenic disorder because it is caused by changes in the gene. So mind you, we are still talking about genomics. Genomics include chromosomes and gene-related disorders. Now, what is epigenetics? What is epigenomics? Epigenomics is a study of how cells control gene activity without actually changing any part of the DNA. There is no change in the DNA per se, but still there is change in the whole function of an organism. So, all the external influences that control the activity of the gene without changing these pearls, without changing these sequences, is known as epigenetics. Now, I will keep on repeating this definition throughout my lecture so that by the end of the lecture, we are clear about what epigenomics is. Genome is related to the DNA, and epigenomics is related to all the changes that occur on the DNA, but they are not caused by any change in the sequence of the DNA. So epigenetics is on top of genetics because it can affect genetics from outside. These are chemical modification on the DNA without altering the DNA sequence. The key here is in understanding the concept of epigenomics is the DNA sequence is not altered. So when we look at this diagram here, the expression of a gene is on here and it is off here. So this can be made on and off by certain changes which are not involved in DNA sequence alteration but other type of changes. Now when we come to the human DNA structure, each cell in our body contains the same DNA which I spoke of as the genome. It is packaged into chromosomes and we all have 46 pairs of chromosomes. All cells contain the same genes, all cell contains the same genome. But what is different in all of these cells? The gene expression, that is conversion of this gene to a protein, is what is difference in each cells, and this gene expression is controlled by our epigenomic signature. So when we talk about how these epigenomics modification work, they actually come together to either tighten the DNA or to loosen the DNA. Once the DNA is very tight, it becomes non-functional, and once the DNA is loose and relaxed, it becomes functional. This modification of making the DNA loose or tight is known as a histone modification. This is the first epigenetic modification that occurs outside and it occurs throughout our life. Now, the second important modification in epigenetics is the DNA methylation. If we look at the bulb here, this is a gene that is being expressed. Whenever a methyl group, that is a CH3 group, is added to a gene, the gene becomes switched off. Any methylation that occurs in our body makes the gene switched off and these genes stop functioning. So we can see here that there is no problem in the sequence of the gene, and there is nothing that is deleted or duplicated. No chromosome is missing or no chromosome is extra, but still the gene will not function, and this is due to addition of a methyl group on this gene. Now, why do these methylations occur? These methylations can occur due to the pollutants that are present in the environment. They can occur due to the stress that we are facing. They can also occur due to certain diets that we are consuming. So these methylations and demethylations are continuously occurring in our body, and they can affect how our genetics and epigenetics is regulated. Now we come on to a very good example of how this epigenetic works. So now let's talk about identical twins. That is a monozygotic twin. A monozygotic twin is obviously formed from the same DNA, but we see that there are still some differences in the behavior, in the personality, even in the looks of monozygotic twins. How do we explain these changes in monozygotic twin? So as we can see here that this is twin A and this is twin B. This is presence of a methyl group which occurred at some time after that zygote was formed. This made some genes on this twin get expressed, and because the methyl group was not present, present is some genes not expressed. So because of the presence of absence of these epigenetic marks or epigenetic signatures, there are differences which are present in human beings who even have the exact same DNA. Again, focusing on the definition, epigenomics is a study of how a cell is controlled without any alteration in the sequence of the cell. So the sequence of the DNA remains the same, nothing is added, nothing is missing, but there are certain marks, there are certain methyl groups which are added or which are taken off those genes, which can alter the expression of the gene, that is, which can decide if the gene will make a protein or that gene will not make a protein. So epigenetic changes are modification to the DNA that regulate whether genes are turned off or on. These modifications are attached to DNA and do not change the sequence of this DNA. Within the complete set of DNA in a cell, all of the modification that regulate the activity of gene is known as the epigenome. I hope with this explanation and the epigenomics word is clear to all of us. Now, what are the implications of this epigenetics on male infertility and what are its implications on female infertility? So there have been several scientific studies that have interrogated different genomic regions and have found that epigenetic changes, including DNA methylation and histone modification, are associated with infertility. For example, if there is some DNA that is present in the tail of the sperm that is too close together, it will not function and the sperm will lose its motility. And if there is some DNA in the sperm which has a methyl group added to it, the gene expression will be switched off. So certain marks can be switched on or certain marks can be switched off, and this can decide the motility of the sperm as well. This association between an epigenetic profile and infertility appears to exist even when the semen parameters are normal. So actually it is quite a challenge to really understand that when can we actually say that the epigenomics is what is regulating these changes. Reading out the epigenetic pattern in sperm DNA presents a particularly interesting opportunity for diagnosing male infertility in cases of an unexplained infertility. As we see in this diagram here, this is the sperm, as we can see. So there are many factors that are occurring which are actually causing the sperm to be troubled or its DNA to be disrupted. This includes diet, smoking, and it also includes the pollutants from outside like ionizing radiation and intake of alcohol. Epigenetic alterations are induced by lifestyle as well as environmental factors like diet, smoking, radiation, and alcohol consumption, and this can have substantial effect on sperm function. As the first consequence, these modifications can induce sperm alterations, leading to an impairment of male fertility. When fertilization even occurs, there are a lot of alterations that still keep happening because of these epigenetic modification. Then there can be alterations in embryo development, there can be congenital diseases birth, even late onset diseases like obesity, hypertension, and diabetes that can be present. So when we talk about epigenomics, we talk about an interaction of environment with our genome, which causes certain genes to not function and finally leading to infertility in the male and due to sperm alteration, as well as early and late onset disease in the offspring. Now coming on to the female, we have we've learned about the effects of pollutants and other toxins on the sperm due to the epigenomic interaction. Now we come on to the female fertility. So DNA marks that are present on the oocytes also could lead to a negative reproductive potential. Several new biological age predictors have now recently been introduced, and I am sure my IVF senior and colleagues here would know this much better. Although the most famous model was the Horvath clock, which said that with increasing age there decreases fertility, this predictor can hardly be used for tissues undergoing hormonal changes as well. Epigenetic changes that occur in the cumulus oophorus, which does not go as the same rate in all women with the chronological age. We have seen that two women who are of exactly the same chronological age will end up having different fertility and different fertility potential. What answers that? That particular phenomenon is what is answered by the epigenomics of an individual. Epigenomics, we can say that to some extent could be in our control, which could be due to our lifestyle and the choices that we make. So even if a person is of a younger age, there is a chance of a low fertility due to these epigenomic modifications, which can be modified by drugs which are being developed lately. So, interestingly, nutrients taken in the diet can have a crucial role in modulation of aging. In fact, it has been demonstrated, for example, an incorrect intake of folate and homocysteine can significantly alter DNA methylation and subsequent epigenetic reprogramming of cells. Now, what I want to talk about here is that we all are giving folic acid 2 months and 2 months before, 3 months after pregnancy, etc. But I think it is important to mention that we need to give folic acid only up to a point that we are suspecting a neural tube defect to develop in. Folic acids, which continue throughout pregnancy, cause a change in the epigenomic mark of an individual. So, because all the 1-carbon metabolism in the body is controlled by folic acid, so we must see that we are not increasing giving too much folic acid as well, because it is changing the epigenomic marks of an individual. So here in this diagram, which is taken from a Lancet journal recently published, which says that vegetable-based diet, polyunsaturated fatty acid, multivitamins and minerals, they lead to a good epigenetic modification, whereas a animal-based diet, carbohydrates, sugar, trans fatty acid, and Western diet caused epigenetic modification, which will decrease the ART success rate. So, in addition to a good IVF cycle, we need to focus on this as well. There is, if all these diets are consumed, there is effect on the genetics, on the epigenetics, as well as on the gut microbiota, and there is a decrease in the ART success rate. There is a risk of ovulatory disorder, infertility, and the live birth rate, as well as the pregnancy rates are decreased. And this is because all the changes that occur in on the DNA due to the epigenetic modification. So again, this is another diagram here which shows maternal exposure to environmental factors, diet, genetics, and parameters such as lifestyle can come together and lead to a decrease in the fertility potential. Now we have finished discussing about epigenomics that occurs in the fertility, and now we come on to what is really being practiced and what is right now the standard of care in fertility. So I think this topic we also covered a part of it in the last talk. Now we talk about male infertility and how much percentage of infertile males have a genetic cause. So the first thing is that 4 to 5% of the males who are unfertile have a genetic cause. And when do we want to test males for infertility? Include a non-obstructive azoospermia, a severe oligospermia, and non-palpable vesa. We do not need to test all men with infertility, and the indications when genetic testing we must not do is when there is a history of a known infection that is known to cause infertility, and secondly when there is a prior birth of this person, he has fathered a child. In those cases we do not need to test for genetic disorders. So now again, the indications include a non-obstructive azoospermia, severe oligospermia, and non-palpable vasa. So the first thing is the congenital absence of vas deferens. If there is a congenital absence of vas deferens, this is most commonly associated with cystic fibrosis, and this is caused by variants in the CFTR gene. When do we think of a CBAVD or a congenital absence of vas deferens? When there is azoospermia and there is absence of vas deferens on palpation, and we do a genetic testing called as NGS based CFTR sequencing, as we can see here, and these men will actually be able to father children because this is an obstructive type of azoospermia, and men with CBAVD typically have normal spermatogenic potential and are therefore excellent candidates for ART. Men with CBAVD should undergo renal imaging to look for associated defects, and partners should also be tested for genetic disorders. Now coming on to the other major category of genetic disorders causing infertility is the Y chromosomal defects. These are microdeletion in the AZFC factor gene, which can lead to a non-obstructive azoospermia. When there is an obstructive azoospermia, we test for cystic fibrosis. When there is a non-obstructive azoospermia, we order a PCR or a MLPA test for looking for Y chromosomal microdeletion. And this test will show us that which part of the Y chromosome is deleted, because of which the sperms are not being able to form. Now coming on to the most common cause of male infertility, and we all know it's the Klinefelter syndrome, in which we know there's an extra X that is present. This is the karyotype of an individual with Klinefelter syndrome, which shows an XXY. So the phenotype of an individual is the male will have tall stature but will have small testes, gynecomastia, and azoospermia, and the test is very easy to perform. It's a simple FISH or a simple karyotype that will give us the diagnosis. The treatment is, of course, early testosterone supplementation. This may not correct infertility, but will give him more of a maleness that he can lead a quality life with. Of course, there are some other causes of male infertility that could start from the pituitary to the adrenal and to the testis and to the sperm. And the test, a combined test for male infertility, if we need to do, would include that we first figure out if it is a obstructive or a non-obstructive azoospermia. Then we look at the semen analysis. The semen analysis will give us the clue on which gene could be involved, and once we know that what we want to look at, an obstructive or a non-obstructive cause, then we can go ahead with testing. For an obstructive cause, we will look at cystic fibrosis gene. For a non-obstructive cause, we will look for Y chromosome microdeletions. So it is where male infertility is a little easier to deal with, because we just have two major groups, a non-obstructive and an obstructive azoospermia. Also, as I mentioned here, depending on the shape of the chromosome of the sperms, the abnormality in the shape, which is also known as dysmorphology, we can test for specific genes. Now, all these genes can be tested together by a test known as exome sequencing. Coming on to what we more commonly see, that is female infertility. The 5 common categories of female infertility include Turner syndrome, which we all know is the monosomy X. Girls present with primary amenorrhea, and the test of choice is a karyotype that we need to do. When we have a girl whose karyotype we perform due to a primary amenorrhea or infertility, and the karyotype shows XY, we then think of a 46XY gonadal dysgenesis, and there are certain genes which are present and which can be detected by exome sequencing, which will give us the answer of this sex reversal phenotype. Then if we find a 46XX karyotype, but the ovaries are very small, and obviously the girl does not have Turner syndrome, this condition is known as a 46XX gonadal dysgenesis. So mind you, in 46XX and 46XY gonadal dysgenesis, the phenotype is perfectly normal. So this is a girl with Turner syndrome. This is the karyotype where there is a missing X chromosome. So girls with Turner syndrome typically have primary amenorrhea, short stature, renal abnormalities, and very specific cardiac abnormality known as a coarctation of aorta. I just discussed XY gonadal dysgenesis when there are phenotypic females with defects in sex determination, also known as Swyer syndrome. And these girls will have primary amenorrhea and, of course, infertility. We can look at these mentioned genes here to find out the cause of their infertility. Then we have XX gonadal dysgenesis, in which this is a female hypogonadism with no functional ovaries, which are present to induce puberty. The karyotype is normal, that is 46XX. This is one of the most underdiagnosed condition because the phenotype is normal, the karyotype is also normal. The only thing that is abnormal is the hormones in the ovary. This is another genetic condition, an autosomal recessive condition, for which we have to look at the genes and perform a exome sequencing. Finally, a common disorder known as a fragile X premature ovarian failure, in which, as compared to this normal X chromosome, there is a problem in the X chromosome in these individuals, and there is presence of fragile sites, which results in a premature ovarian failure. And the test we need to do here is a fragile X PCR. So finally, in female infertility, we do a fragile X PCR, and then depending on the phenotype of the patient, on looking at and examining the patient, we decide whether to do an exome sequencing or not. And this is all following a conventional karyotype. So first step is a karyotype, then followed by a fragile X PCR, and then we need to decide we need to do exome sequencing or not. So to end my discussion, I would like all of you to maybe go through this really interesting paper of epigenetics in the uterine environment, how maternal diet and ART may influence the epigenome in the offspring and also cause long-term health consequences. We can talk to our patients about this, tell them that how important is the role of their diet and their environment in shaping their child's future as well. With that, I would like to conclude and say that epigenetics is the effects on the function of the cell which do not alter DNA sequence. Diet, smoking, alcohol all modified the epigenome and are being increasingly implicated in infertility. Still, epigenomic testing we are not doing, but genetic testing is the standard of care and should be offered to infertile couples according to the indications mentioned. Thank you so much.